Bonjour tout le monde, mon nom est Raed El Ferjani, je suis agent d'échange de connaissances et de mobilisation autochtone au Centre de foresterie des Laurentides et j'animerai ce webinaire. Avant de laisser la parole à mon collègue Armand Séguin qui va présenter la conférencière, voici quelques consignes d'usage. Au bas de votre écran, veuillez fermer votre caméra si ce n'est déjà fait. Pour activer la boîte du chat, cliquez sur l'icône bulle en bas à droite. La période des questions se fera à la fin de la présentation. Finalement, j'aimerais mentionner que le webinaire est enregistré. Je laisse maintenant la parole à Armand.
Alors, bonjour tout le monde, mon nom est Armand Séguin, je suis co-directeur de la division de biologie forestière au Centre de foresterie des Laurentides. Alors, bienvenue à ce webinaire de la 38e saison des colloques du CFL. Donc, la conférence d'aujourd'hui s'intitule Genomics-driven monitoring for genetic conservation and breeding in trees. Et c'est avec plaisir que je vous présente la conférencière, Mme Jill Hamilton. It's a pleasure for me to introduce your guest speaker, Jill Hamilton. Madame Hamilton est professeure associée à l'Université d'État de Pennsylvanie au département des sciences et de la gestion des écosystèmes. Elle est aussi directrice du Schwartz Center for True Molecular Biology et titulaire de la chaire Iverson de recherche en sylviculture. Donc, elle a une formation tout à fait canadienne où elle a obtenu sa maîtrise à l'Université de Queens et son doctorat à l'Université de Colombie-Britannique. Le sujet de sa thèse portait sur la base génomique de l'adaptation dans l'hybridation naturelle entre l'épinette de Sitka et l'épinette blanche en Colombie-Britannique. Elle a occupé 2 postes de recherche postdoctorale, un à l'Université d'Alberta, où elle a étudié l'écophysiologie de l'épicéa dans diverses conditions environnementales, et l'autre à l'Université de Californie à Davis, où elle a étudié les bases génomiques de la préadaptation. Donc, elle a officiellement débuté sa carrière en enseignement à l'Université d'État du Dakota du Nord en 2015 et rejoint l'Université d'État de Pennsylvanie en 2021. Donc, elle est aussi actuellement présidente de la Western Forest Genetic Association. Elle a reçu déjà du financement de différents organismes subventionnaires, le NSF, l'USDA et le US Forest Service, donc pour soutenir ses programmes de recherche. Donc, je vous souhaite un bon colloque et je laisse maintenant la parole à Jill. Hand the floor over for you, Jill. Thanks.
Okay, thank you. Thank you so much, Armand. That was a really lovely introduction. Admittedly, my French is a little limited these days, but I caught most of it. So thank you so much for the invitation today, everyone. And I really appreciate the opportunity to share some of the research that we have ongoing at Pennsylvania State University, where we're thinking about genomics-driven monitoring for genetic conservation and for breeding in forest trees. And I'll admit, I have the wonderful opportunity to be the director of the Schott Center for Tree Molecular Genetics here at Penn State, and I'm the Iverson Chair of Silviculture Research. What does this actually mean? Well, it means that I get to work with an outstanding group of young researchers that are really passionate about contributing to conservation and restoration in a changing climate. So the work that I'll be presenting here today really reflects this team effort. And to introduce you to my crew, we are what we refer to as a Muddy Boots crew. Essentially, this means we use a combination of field observations, field experiments, environmental modeling, and genomic data to understand traits that are important to adaptation to climate, climate-related stresses, and diseases. And we wouldn't be able to do what we do without collaborative partnerships. I think really key to conservation and restoration is our collaborative partnerships. And I want to acknowledge our various funding sources and partners that have assisted and continue to assist in a myriad of different ways to the research programs that I'll be talking about today. So as Armand mentioned, I joined Pennsylvania State University in 2021. And when I first arrived here, I was taken out to this common garden experiment because I am a lover of common garden experiments. And this is a picture that I took at the common garden site. Today, this common garden fascinates me as it represents something that is perhaps a little sad, but also perhaps a little hopeful. In the 1970s, Dr. Kim Steiner established a beautiful range-wide collection of green and white ash at this site, which is 5 minutes from my office on campus here. And it was a luscious and productive common garden experiment representing provenances across the range. However, during the eruptive invasion of the emerald ash borer, nearly all of the green ash trees died. And over a period of approximately 6 years, 99% of the green ash died and 87% of the white ash died. And in fact, today, none of the genotypes that were initially planted remain living today. And this strikes me as pretty sad, as the emerald ash borer has decimated ash species across eastern North America, and it is continuing to expand its range. As one of the most destructive pests ever introduced to North America, the emerald ash borer has wiped out millions of ash trees across the eastern US and is marching west and is marching north, and it has now been found on the West Coast, where it has been identified just outside of Portland, Oregon. However, despite these impacts, there is hope, I think, because there is what we see evidence of interspecific and intraspecific variation in susceptibility to EAB across different ash species and across populations within species. Even in this common garden experiment where you can see the standing dead, Dr. Steiner noted that measures of EAB injury were significantly influenced by genetic effects, site quality, and the neighborhood of the pests. So while there was near complete destruction of these trees, genetic and site effects moderated the rapidity with which trees were injured and killed, suggesting that there is variation in susceptibility, and where there is variation, there is evolutionary potential. So when we think about species' ability to adapt and respond to changing conditions, we often think about the ability of species to adapt and their evolutionary potential. A species' evolutionary potential will be tightly influenced by both the amount and distribution of genetic variation important to adaptation. In addition, it will be influenced by demographic variation via population vital rates and changes in selective pressures through stochastic environmental fluctuations or changes in abiotic and biotic stresses. In species such as ash, which are often keystone within their native distributions, preserving intraspecific and interspecific population variation for different ash species will be essential. In essence, this safeguards the genetic variation, that raw material upon which natural selection acts, and the evolutionary potential of the species needed to establish foundational resources for breeding and restoration. Interestingly, despite the growing emphasis on the importance of genetics in conservation and the value of intraspecific variation to the maintenance of species' evolutionary potential, global conservation policies and actions have largely neglected protecting and monitoring genetic diversity. Indeed, interspecific variation, again, that raw material upon which natural selection acts, is facing a hidden biodiversity crisis. Recently, the IUCN suggested that the rate of loss of intraspecific variation is many times that of species loss. And we can see that reflected here in this recent perspectives piece by Simone DeRoche, where we see a comparison between historical and contemporary estimates of heterozygosity based on 88 different studies, 91 different species. And if we compare the amount of genetic diversity lost or gained across time, we see on average that losses are much more common and dramatic than gains per species of conservation concern. For forest trees, this is particularly important, as many forest trees at risk today have broad geographic ranges and exhibit substantial genetic variation, but also exhibit often strong signatures of local adaptation. So if populations are lost, there is a high probability that that adaptive genetic variation may be lost forever. So with an overarching goal to maintain an ability to cope with change, our group thinks a lot about how different evolutionary mechanisms, including selection, gene flow, drift, contribute to exchange and maintenance of genetic variation needed for adaptation. We think about this particularly in terms of how we design and implement conservation collections, how we transfer seedlings or seeds for restoration, and how we approach conservation of rare and endangered species. Ultimately, the questions and approaches that we need for conservation and restoration of forest trees requires an interdisciplinary approach that spans the lab, the field, and the data science nexus. And so we leverage a variety of tools, including population genomics, quantitative genetics, and spatial modeling, among others, to ask the question, how does genetic and environmental variation influence the expression of traits important to adaptation, where ultimately our goal is to understand these relationships to make timely and informed decisions for conservation and restoration, particularly in the context of changing climates and in response to exposure to new and expanding biotic threats. So today, I wanted to discuss opportunities where genomic screening alongside ex situ conservation programs have value to breeding, and discuss different considerations that will be critical to establishing future supplemental ex situ collections needed to preserve the long-term evolutionary potential for species, and how this information can inform breeding long-term for resilience. Given the need for proactive conservation, an assessment of genomic variation within and among ash populations will advance our understanding of neutral and adaptive processes and how they have shaped species distributions. So today we'll be taking a genomics approach to asking how have different evolutionary processes influenced standing genomic variation for ash populations. The development of these genomic resources may have major impacts on the success of future breeding and restoration programs. I'll also be asking the question, can we optimize preservation of evolutionary potential for species of concern? There's a lot of data that exists within ex situ collections. Can we use this data to identify gaps in our collections and then coordinate efforts to optimize our collections across agencies? And finally, I'll talk very briefly about our long-term goal, where we really want to leverage an understanding of genotype to phenotype across environments for conservation and breeding, where the resources that we can establish linking genotype to phenotype will provide a foundation for future comparative work directly applicable to state, provincial, and national forest health initiatives. So when we first started our research program on ash, one of the things that we were really interested in doing was creating a genomic passport for germplasm. The idea behind this has been well established in many agricultural species. However, increasingly, its application in conservation is leading us into new areas. One of our goals is to establish genomic passports for our ex situ and our living collections for species at risk. In essence, we're pairing genomic monitoring with our ex situ and living collections, creating this genomic passport of the maternal lineages that are the foundation for seeds or seedlings that will be used in research, restoration, and breeding. And so when we initially got into this, our thinking was to be very proactive in pairing genomic passports for Oregon ash, where at that time, the emerald ash borer had not yet hit the West Coast. And the genomic passport essentially includes a two-pronged approach, working with many, many, many collaborative partners. We took an approach where we collect leaf tissue, and essentially this leaf tissue maintains a record of the evolutionary processes that has shaped the genetic structure of populations in the wild. Essentially, those populations that natural selection has acted upon. Then we sometimes, if possible, we compare that with a genomic assessment of our seed collections. And that provides us the opportunity to compare variation preserved with populations on the landscape and to understand how the collection process itself, which occurs across space and time, may impact variation preserved ex situ. Essentially, this allows us to ask the question, are we preserving what we think or want to be preserving? So we have been contributing to leaf collections and seed collections across multiple Fraxinus species. These data can assess standing genetic variation today, identify genetic clusters, and identify those factors influencing clustering. Using these data, we can target supplemental collections, identify genetic clusters or progeny associated in those genetic clusters critical to assessing in an EAB resistance breeding program, and identify how the environment may influence adaptation for long-term restoration planning. Long-term, our goal is to ensure that with conservation actions taken, we minimize loss of genetic diversity and ultimately maintain genetic variation needed so that populations can adapt to change, thus preserving species' longer-term evolutionary potential. And we've been doing this for a while now in 4 of our favorite Fraxinus species. We initially implemented this program in Fraxinus latifolia as a proactive conservation program, and now we're complementing it with range-wide landscape genomics approaches in Fraxinus pennsylvanica, Fraxinus americana, and Fraxinus nigra. Today, I'll be focusing on some of our preliminary data from Fraxinus latifolia, so you'll have to stay tuned on our eastern ash, but that will be coming soon. So, as part of this project, in Oregon ash, 61 populations were sampled across much of the range of Oregon ash. In total, nearly 1,000 individuals were sequenced using a dd-rad-seq approach, and following de novo assembly and stringent SNP filtering, we established a data set of 42,000 SNPs to assess standing genomic variation. And I will say, this is not possible without the help of volunteer collectors from the U.S. Forest Service, the Oregon Department of Forestry, BC Forest Service, Washington Department of Natural Resources, Huntington Botanic Garden, and many, many, many more partners. There was a call put out requesting help in leaf collections and seed collections, and in requesting help in identifying sites where we might actually contribute to collections. And our partners across the range answered the call. So when he joined my group, Dr. Anthony Melton was really excited to dig into these 42,000 SNPs across 61 populations to examine range-wide genomic variation in Oregon ash. And the first thing that he did was he performed a principal components analysis to assess differentiation and variation across the range. And interestingly enough, we saw substantial differentiation in two groups along our very first PC1 axis, which you can see here, where we've got a number of populations which all come from Southern California, grouping together on the right, and we've got what appears to be a latitudinal cline with BC poking up at the top along the top of the PC2 axis. So we were really interested to ask, what is going on with these Southern California populations? And so I will take a little brief tour of what is going on, but you're going to also have to stay tuned for more. What we're finding is that there is a complex evolutionary history in our southwestern ash species, but Anthony was able to assess potential for genome duplication in our southern populations. And what he identified right away was that our southern populations that are genetically divergent appear to be tetraploids. So there's some evidence of polyploidy in the southwestern Oregon ash populations. We are currently digging into this more thoroughly. We're also looking into the potential for hybridization with Fraxinus velutina in this area, but what we're finding is in the southwest, the Fraxinus populations are very dynamic, and we're excited to dig into this more complex evolutionary history and evolution of polyploidy within this system. So that's a very brief divergent into the fact that polyploids exist in Fraxinus, and they exist in southwestern Fraxinus of Oregon ash. But what we find on average is that we do see diploids from Northern California all the way north to British Columbia. And so when we subset out those southwestern California populations and just look at that one big group that was identified in the first principal component, we begin to see more structure in our populations. And now what we've done here is you can see PC1 is actually on the y-axis and PC2 is on the x-axis. And we've done this specifically because I want to kind of indicate that we've got a latitudinal gradient here in genetic structure, where we've got our British Columbia populations in the north, high up there on the PC1 axis, and we've got our California populations on the bottom of the PC1 axis, with a nice gradient of individuals in between as we move from California through Oregon, through Washington to British Columbia. So one of the first questions that Anthony and I were interested in asking was, how have different evolutionary processes influenced the standing genomic variation for these Oregon ash populations? And again, this is now, we're just looking at from Northern California all the way up to British Columbia. And what we find is that when we look at genomic structure of these populations, we have evidence of 4 genetic clusters. And we have to ask the question, well, what might be influencing the clustering of individuals? What evolutionary processes, considering gene flow, genetic drift, and selection, might be influencing the structure across the species range? And can we use that understanding of structure to impact restoration and breeding programs? And so what we did find immediately is that the British Columbia population actually shows really low levels of nucleotide diversity. This suggests that perhaps this population was founded with a subset of genetic variation across the range and may have experienced some drift and isolation since that may be contributing to it being a unique genetic cluster. But what else is going on across this range? Well, we see that isolation by distance suggests patterns of gene flow are associated with post-glacial recolonization that has influenced genomic structure. Geographic distances associated can predict genetic distances. But we know as well this region is influenced by changing environmental conditions. And we see that isolation by environment as well suggests that the environment has influenced the accumulation of genetic differences. So thinking about this together, when we use a variance partitioning approach, we see that geography and climate explain a large proportion of the among population genomic variance for this species. This is perhaps not surprising, but tells us the degree to which we should be thinking about this in the establishment of progeny trials. To dig into the different evolutionary processes a little bit more, we can dig into things like gene flow. And in this figure, what we're showing is a visualization of estimated effective migration surfaces, where we're trying to visualize potential connectivity across the species range. And where you see indication of blue indicates a higher probability of connectivity, and colors indicated in brown indicate areas where we see on average lower levels of connectivity and potential barriers to gene flow. And what we see in the system is that on average, we see higher levels of connectivity along river valleys. This makes sense given the species biology. It's often found along river systems. And so throughout the Willamette Valley, we see higher migration rates for individuals, suggesting substantial connectivity throughout these regions. We do find some evidence that there is reduced connectivity in some regions that are associated with higher elevation, such as that in Northern California in the Pitt River Valley, where we're seeing lower levels of potential migration. But overall, this tells us that there is differential connectivity across river valleys influencing genomic structure of these populations. In addition to this, we know that natural selection has influenced the genetic structure of these populations. So using an RDA approach to test for genotype environment associations, we identified a number of environmental variables that contribute to genomic structure. And here again, we've got the RDA1 axis along the y-axis and RDA2 along our x-axis. What we can see is that the primary contributor to RDA1 is both mean annual temperature and relative humidity, whereas RDA2 is strongly influenced by this continental gradient and frost-free period, where this continental gradient, that TD value, really reflects the fact that some individual populations are sampled right along the ocean in highly continental climates or highly maritime climates, and we move to more continental climates towards the interior. So understanding how these climate factors contribute to genetic structure is important because this may be important to understanding how they may respond to climate into the future. And so Anthony performed a genomic offsets analysis using 5 informative climatic variables. And this allows us to estimate how much genetic change will be required to adjust to predicted climate change scenarios. And here we're using an ensemble from 2041 to 2070. This is an ensemble that is probably most likely climate change scenario. And what we can see from our genomic offset predictions, it suggests that climate change will likely have a negative effect on genomic diversity of Oregon ash across its range, as the genomic distances are predicted to increase for all populations. Interestingly, we do see lower genomic offsets in the southern distribution of the range and higher genomic offsets in the northern distribution of the range, suggesting that the northern populations may be at risk into the future. And so thinking about this in terms of how climate change will interact with the emerald ash borer now that it has arrived on the West Coast will be particularly important. So as we moved from the past to present with our genomic data, we begin to have an understanding of how different evolutionary processes have influenced standing genomic variation for our ash populations. We can see that gene flow and natural selection have played important roles shaping the contemporary genomic structure of Fraxinus latifolia. This is perhaps not surprising, but what we see is that connectivity and genomic structure are not uniform across the species range, which may point towards regions to target to establish progeny trials, particular genotypes to include in progeny trials for EAB resistance breeding, in regions where we may want to evaluate genotype by environmental interactions in response to EAB. Ultimately, this data is really important because we begin to see the expected projections associated with genomic offsets and how they may impact populations in a changing climate. And we can assess whether these potential changes will be exacerbated by the expansion of EAB on the West Coast. And so one of the ways that we're growing this program as part of our Trees in Peril project funded by the Nature Conservancy, we are expanding upon the work and looking more closely at range-wide landscape genomic variation in eastern Fraxinus species. This is work largely led by PhD student Kira Lopiccolo. And what we're finding is that there's much that we don't know about the evolutionary history of these species, their genomic structure, and the evolutionary potential of our eastern ash species. And this work will provide a foundation to complement existing and expanding breeding programs targeted at developing EAB resistance. So in addition to thinking a lot about landscape genomics and genomic screening for evolutionary potential, one of the things we've been asking is, can we optimize preservation of evolutionary potential for species of concern in our collections? And traditionally, conservation of rare species has included a number of different strategies to preserve biodiversity, including preservation of species in situ where possible through the establishment of parks or reserves, or it may include establishing ex situ collections of a species to preserve that germplasm outside a species' natural range. Ex situ collections aim to collect that germplasm for rare species, agricultural species, and foundational species that may be at risk. And these seeds really reflect the genetic libraries available at that space and at that time produced by that maternal lineage. Data within these ex situ collections are incredibly valuable, and I think that they can provide us a lot of information on how we might supplement existing collections, particularly where our understanding of the distribution of variation between populations, within populations, and even among maternal families may may be unknown. And so using our ex situ collections, we can establish proxies for evaluation of genetic variation across space and time. So I'm going to transition a little bit to another species that we've been working on. We've been working in Torrey pine for quite a while now, and Torrey pine is a great study for the value of ex situ collections. Torrey pine is one of the rarest pines in the world. It's a species where the combination of in situ and ex situ conservation efforts is being paired with genomic monitoring. Torrey pine is often a poster child for rarity in forest trees, where it exists in Southern California and is a charismatic megaflora of the West Coast that exhibits an extremely restricted distribution and limited population size. Torrey pine has thus far benefited from extensive in situ conservation, where 100% of its range is preserved by the National Park Service on Santa Rosa Island and by California State Parks for the mainland population. In addition, there have been multiple efforts to preserve germplasm ex situ across the two existing populations. One of the questions that we wanted to ask, this was when we did not have any genomic data, was can seed morphology be used to quantify variation preserved ex situ? We wanted to take x-rays of our seeds and see if we could take morphological measurements of our seeds and actually use that data in identifying the effort needed to contribute to supplemental collections. And so to do this, we sampled a number of maternal families across the Torrey Pine State Reserve and on Santa Rosa Island. And I had amazing undergrads and graduate students, Lionel DeSanto, Monica Poljar, and Storm Neese, contributing to measurements across these seed x-rays. And what we saw immediately was that you could clearly differentiate the island from the mainland based on seed morphology alone. This was actually really interesting because a previous genetic study at that time based on microsatellites had suggested that there was essentially no variation within either population. And our morphological data suggested that there was actually probably there was definitely differentiation between populations and that there was variation within populations across maternal families. And so this was actually really exciting. So we knew that we had variation within these populations that we wanted to ensure we were conserving. And indeed, when we took an evaluation of all of our seed morphological traits, which you're seeing the different traits on the y-axis here, we can partition that variation attributed to either population differences, maternal families, or individual residual variation. And what we see is there's substantial variation in maternal families, suggesting some family structure. And this is important because we can leverage that information to model what we might need to sample in addition to ensure we're capturing variation in our collections within this population. And so often a goal in ex situ collections is to ensure that we're collecting 95% of the trait variation in a collection. And so we wanted to estimate using our morphological data of maternal families if we could model this. And so what we did was we estimated the number of unique seed trait values captured in a sample of maternal families, our N sub C value here, relative to the total number of unique seed trait values present in a seed population, the N sub T here on the y-axis. And we asked the question, if we increase the proportion of maternal families sampled, when do we hit 95% of the morphological trait variation captured? This is based on existing collections, so it doesn't include collections that we haven't captured. But what it does suggest is that to supplement collections, we need to sample 54 families out of 80 to ensure we're getting 95%. And it gives us a value of potential effort required to ensure that we're collecting 95% of that seed trait variation. So this morphological data without any genetic data at all can be incredibly valuable in actually assessing the degree of effort required to supplement existing collections. In addition to this, we have had the opportunity to perform genomic analysis of ex situ collections alongside wild populations. I'm going to veer again into a new system where we had the opportunity to work in Helianthus maximiliani, a wild sunflower of the Great Plains that is commonly used in restoration. And we wanted to take a genomic approach to compare wild contemporary remnant populations with genomic variation preserved from ex situ collections that had been collected in the 1980s by the USDA in the same region that we collected our contemporary collections. We also wanted to compare that with commercial seed sources used for restoration in the same region and compare that with a population of Helianthus maximiliani that was selected for particular agronomic traits. In this study, basically, we want to ask the question, using genomic approaches, are we preserving what we think we're preserving with our ex situ collections, where we can assess the genetic variation in collections using genomic approaches. And so what we see when we compare across these different seed sources is that the effective population size is actually quite a bit lower in our ex situ collections. It's very low, in fact, relative to the commercial populations, similar to that in the wild populations, but the wild populations are more variable. And this was really interesting, and we didn't expect to see this. So we wanted to ask the question, well, why do we see such low effective population size in our ex situ collections? And it turns out when we take our genomic data to evaluate the relatedness now on the y-axis of our different seed populations, we can see that the relatedness in the ex situ is quite high relative to the other populations, particularly wild and commercial. The commercial populations reflect a bulk collection of seeds, and the wild contemporary populations reflect population sampling. But we think the ex situ collections, while there were a number of efforts made to conserve genetic variation in Helianthus maximiliani, it could be that only few maternal lineages were collected, but many seed heads from an individual were collected to contribute to the relatedness that we're seeing in our genomic data. So this maybe begs the question, do our ex situ collections preserve the genetic variation that we think they're preserving? So a question that we're asking is, can we optimize preservation of evolutionary potential? Well, I hope I've convinced you that there is value in morphological data taken from collections, and this may be proximate variants that we can use to capture supplemental collections ex situ. Where possible, we can pair genomic data in ex situ collections with wild populations, and this may inform future conservation collections as well, where we can identify gaps and maybe how to fill them with respect to genetic variation preserved. And finally, there are lots of opportunities to work with conservation managers to optimize the preservation of genetic variation ex situ, because this will be critical to long-term restoration projects. And as I mentioned, we're continuing to work in this in ash, where key to conservation and restoration of our ash species today really reflects collaborative partnerships across borders, across state lines, across provincial lines, where we need to preserve the genetic libraries of our ash species. We're currently working with several partners, including the National Tree Seed Center, USDA ARS GRIN, and US Forest Service, and many volunteers, largely led by postdoctoral fellow Melissa Lehrer and undergraduate Lucy Anderson, to preserve the genetic library of ash. They're currently taking an approach where they have x-rayed all the seeds coming in, and we are evaluating maternal tree variation. We're assessing damage. We're really excited to learn more about these different maternal seed families that we have been provided. And the real value that I'm excited for is we're continuing to test for adaptive trait differences in black ash, where we're leveraging these ex situ collections to establish living collections or provenance trials to ensure the preservation of material needed for longer term breeding. And so we've been the beneficiaries of range-wide collections, and we'll be planting little ash seeds very soon. And so I hope perhaps I've suggested that for the future, as we begin to leverage our understanding of genotype to phenotype across environments for conservation and breeding, that these replicated common garden experiments that are established through these ex situ collections may provide a foundational resource for breeding and restoration. And I'll be happy to talk more, but stay tuned to hear more about our upcoming emergence of black ash seedlings and the establishment of potential opportunities to plant a regional common garden of black ash. So I hope I've suggested today that from the past to the present, genomics can be useful and be used to inform effective conservation collections and breeding program design. The morphological data or genomic data that we have associated with our collections can be valuable to predicting evolutionary potential of our restored material. And ultimately, these common garden experiments can be foundational to creating material for restoration efforts. And ultimately, as I said, this isn't possible without an amazing team. I like to acknowledge Anthony Melton, Kira Piccolo, Melissa Lehrer, and Lucy Anderson as key contributors to some of the work presented today. In particular, thank you to our funders and collaborators, the USDA McIntyre Stennis Program, the Nature Conservancy, US Forest Service, Canadian Forest Service, National Tree Seed Center, USD GRIN, Oregon Department of Forestry, Adventure Scientists, Huntington Botanic Garden, and so many people along the way. Thank you.
Thank you so much, Jill. Merci beaucoup. Pour ceux et celles qui se sont joints à nous tardivement, je me présente, Raed El Ferjani, et j'animerai la période de questions. Avant de débuter, j'ai des messages d'intérêt et quelques règles d'usage à partager avec vous. Vous pouvez maintenant activer votre vidéo si vous le désirez. Si vous désirez obtenir une attestation de formation continue, notamment en lien avec le règlement sur la formation continue obligatoire des ingénieurs forestiers du Québec, l'unique façon est de compléter l'attestation de participation avec le lien URL que vous trouverez dans le chat sous peu. Veuillez noter que le code d'activité est maintenant intégré. Il y a également un formulaire pour les arboriculteurs certifiés, membres de la Société internationale d'arboriculture Québec, qui souhaitent cumuler des crédits de formation. Vous pouvez poser vos questions dans la langue officielle de votre choix Et il y a deux façons de le faire, verbalement en cliquant à droite de votre nom dans la liste des participants et sélectionner l'icône main levée. Lorsque je vous nommerai, vous n'aurez qu'à enlever votre mode sourdine en cliquant sur l'icône micro. Après avoir posé votre question, s'il vous plaît, vous remettre en mode sourdine. Nous souhaitons avoir une question à la fois, autant pour donner la chance aux conférenciers de bien répondre à votre question et de laisser la chance à tous de poser leurs questions. Si vous préférez poser votre question par écrit, utilisez la boîte du chat, sélectionnez l'icône en forme de bulle en bas à droite pour l'activer si ce n'est pas déjà fait. Nous vous demandons de vous identifier clairement en donnant votre nom et l'organisation que vous représentez. There are two ways to ask your questions, verbally by clicking to the right of your name in the list of participants and selecting the raised hand icon. When I name you, remove mute mode by clicking on the microphone icon. After asking your question, please click back on mute mode. If you prefer a written question, you can use the chat box. Select the bubble icon and the bottom right to activate if it's not already done. We ask you to clearly identify yourself by giving your name and the organization you represent. La période des questions est maintenant commencée. Our guest speaker can now answer your questions. On a une question orale de Mélanie Zakaria.
Yeah, thank you very much for this nice talk. Yeah, my name is Melanie Zacharias, and I'm from the Université Laval in Quebec. And I have a question regarding like the first project you shared about a genetic clustering and the polyploidy. And I'm working with Aspen, and I also see different polyploidy levels in different genetic clusters, and especially polyploidy in the Mexican population, which experience more drought. So my question for you is if you have also the impression that like drought stress favors polyploidy, and what you think could be a possible explanation.
That is a really good question. I don't know if we know yet if polyploidy is contributing to adaptation to slightly more extreme environments. I think I might hypothesize, however, that there's got to be some advantages to having a genome duplication event, because having a genome duplication event is costly. That it requires a lot more energy for a plant to be a polyploid. So if that does allow them to persist in slightly more drought-stressed or water-stressed environments, that seems a reasonable scenario. In our case, we're interested. We don't know yet if the polyploidy is due to allopolyploidy or autopolyploidy. It could be a complete duplication, and in that case, it's an autopolyploid individuals. But it could be an allopolyploid where you have the combination of two genomes being maintained from different species within an individual. And in this case, the allopolyploids may have the advantage of having adaptations associated with a latifolia parent. And in this case, we predict that the allopolyploid, if it is allopolyploid, it would be a Fraxinus velutina parent. And in this case, you'd maintain the adaptations of the two within an individual. So there could be advantages, but there are probably some costly trade-offs to being polyploids. But I'm very excited to kind of explore these questions further.
Thank you.
Thank you. So do we have another oral question? Nathalie Isabelle.
Hello, Jill. Very an excellent presentation. Very interesting. So people, yes, the genetic diversity is so important, and sometimes we just forget about it when we discuss about biodiversity. I would like to know, do you think that we may observe something similar on the eastern coast, because there are so many ash species, more than perhaps on the west coast? So because I don't remember there are many Fraxinus species in Europe and Asia, so I don't know. I haven't thought about polyploidy in ash, so I was wondering if you think that there might be the same sort of polyploidy.
Yes.
Okay.
Short answer, yes. What we do know right now is that, for example, Fraxinus americana, it can be a diploid, and I think it can be a hexaploid. And there is a version of Fraxinus moriana, which is within the Americana clade that is hexaploid. So we have some evidence already that we're going to come across polyploids in the eastern Fraxinus species. I'm unsure if we'll come across it in Fraxinus nigra, given the evolutionary divergence of the nigra lineage, but I suspect there could be evidence of it in Pennsylvanica or Americana. I also have a suspicion that there is probably some hybridization between Pennsylvanica and Americana on the East Coast, and I'm very excited to get into that data soon. But we are now complete our DNA extractions for those range-wide samples, so we're going to be sending that out for genomic library prep and sequencing pretty soon. So we'll have some more answers soon.
Okay. Thank you.
On a une question de Armand.
Yeah, very interesting talk. I have a very specific question, though, is about the in terms of when you collect seeds, you observe in ash, essentially, you observe a difference in terms of germination rates and stuff like that. Is that from the real conservation aspect? I mean, is that really a limiting factor? And I guess you have done a little bit of that with all the collection for extracting DNA or things like that?
Yeah, so we are just getting into germination trials right now for our black ash range-wide collections, and I will know more of that. We're going to be germinating our seeds probably in about a month and a half. But we really don't know. In all honesty, we're tracking the timing of when the collections were taken, the year that the collections were taken, and we hope to use our understanding of the climate data of that year to associate with germination success. But I think that will be very critical going forward for these collections, which really advocates, I think, when doing these ex situ collections, to consider both time and space. So ex situ collections may not be one and done. It's probably to ensure that we're capturing variation and the ability for many potential germinants over time. We may have to go back and recollect to ensure we're preserving the genetic variation that we want to preserve.
Thank you. Very interesting.
Thank you, Jill and Armand. We have a question from Chedli Castalli.
I think you're muted.
We don't hear you, Chedli.
Okay, can you hear me now? Sorry.
Yes.
Okay, so thank you for the nice presentation. And you mentioned at the beginning that since there is some genetic variability in the ash tree in terms of susceptibility to the emerald ash borer, that there is some hope that there will be, or that there is some potential for evolution. But then I wonder if you have any thoughts about, okay, if a pool of the trees survive, so a pool of resistant trees survive, like obviously you will have lost some genetic variation. And then do you have still any hope if there is another pathogen or another insect or like, or another crisis? Like, what do you think about the next thing that might happen?
The idea of repeated kind of bottlenecks is definitely a scary one, and I think it is something many of our forestry populations are experiencing. I think that one of the reasons that I'm really excited to leverage the ex situ collections to establish provenance trials is to ensure that we're creating some programs, like if as we continue to breed for EAB resistance, we are going to reduce genetic variation in our breeding populations. But if we can go back to these common garden experiments and provenance trials and leverage those in addition to EAB resistant bred populations, then maybe we are still preserving some range-wide variation that will be critical to adaptation. But it is a scary perspective, repeated bottlenecks.
Thank you, Jill. Nathalie, oui, une autre question?
Yes, I just forget to mention that I'm Nathalie Isabelle. I work for Natural Resources Canada. Actually, I'm wondering what would be your advice, considering like climate change on the top of that. What would be your advice? Because for many species, we've realized that we don't have any baseline data in terms of genetic data, the population structure, and what is there in terms of standing genetic variation or population exchanges. So what would be your advice to people like policy analysts who have to do some triage in terms of decision, because it's all the time it's about the question of money, you know. But I realize that for ash, perhaps we have waited a bit long before doing something. So I'm just curious to hear what would you would be, considering your experience in genetic conservation. What would be your advices?
Oh, that's a tough one.
No, but you know, it's
no, you're right. I think with, say, as black ash as the example, what we don't know anything about the genetic variation existing today in black ash. We know that those populations are disappearing. So we have an understanding that the environment does influence genetic variation range-wide for species. So we can leverage our existing environmental data to target regions for sampling ex situ to get that seed ex situ in the bank right away, and develop those partnerships with people that are performing those collections. Because I've been incredibly hopeful by the number of people that wanted to act immediately with the Oregon Ash project, that they wanted to perform the seed collections and get those collections in the bank and provide us information on sites to visit to supplement collections. So I think there's community-based approaches that we can leverage to ensure that these collections are happening today. We can use environmental data to inform, to target collections. But yeah.
And in terms, like I just want to complement, perhaps I just as a case study, for maple, actually we have almost nothing on maple, which is like a kind of emblematic tree here in Eastern Canada. So it's I think that there should be we should go and perhaps do some triage and try to get as much as and develop those resources for ex situ collection for seed. But seed it was not was not really popular at some point. But right now we realize that we may have some problem of of seed production in the future due to climate change, because we observe the shift in leaf phenology, but we might observe also a shift in reproduction. So, anyways, I don't know how we should prioritize the species. Should we think in terms of what's the next invasive species that's gonna come, or we need to think in terms of what should we do first.
Yeah, I think there are different lists of species that may be listed as in the context of climate change winners, climate change losers, and those that may not be as impacted by climate change, and combining that with the impact of existing biotic threats and expanding biotic threats. I think the list is increasingly long, that are really requiring some imminent resources towards their conservation, and something as emblematic as sugar maple. I mean, collections, while they are time consuming and require timing and effort and people, I think those are a great first bet to start.
Okay.
Yeah.
Okay. Thank you.
Merci. On a une question de Pierre-Alexandre.
Good morning, Jill. Do you hear me?
Yes.
Yes, okay. I'm Pierre-Alexandre, a student of Nathalie Isabelle, working on the genetic variation in black ash. And I was curious to know, how do you plan to establish a black ash common garden in presence of emerald ash borer?
This is a good question. I have not quite resolved this yet. So we will be germinating them in our greenhouse this spring, and we hope we would like to be able to plant them out. The emerald ash borer in our region is not at the high levels that it was. So if emerald ash borer impacts the trees, and they probably wouldn't at a young age, but they would probably hit at a more seedling to advanced age, then there may be opportunities to actually treat the trees in common garden experiments so that we can maintain the trees.
Thank you. That's a good idea.
Thank you. Est-ce qu'on a d'autres questions orales parmi les participants? Do you have other oral questions to Jill? Oui, Nathalie.
I have a lot of questions. Are you aware? But I don't know if there is any question in the chat, because I don't want to take the place of another person.
Non,
Okay.
pour l'instant, on n'a pas
Okay.
d'autres demandes de questions.
So I was wondering, do you know, Jill, if there was any study looking at the if the environmental impact, or if extreme events have, I would say, precipitated or increase the rate of the speed of the infestation of emerald ash borer. So, because I know in US the EABs has been there for a long time, so I was curious to know if you are aware of such a study, or if we don't have any of those data.
I'm not aware of a study in that regards. I think my impression is it came very quickly and fast, and went through very rapidly. And my impression was that, like in the Common Garden here at Penn State, which was a range-wide collection, yes, the invasion went through over a period of 6 years, but which is pretty rapid to take out an entire range-wide Common Garden experiment. I don't know that there were other stresses at that time, particular stresses that would have exacerbated the effects. I could imagine a case, and we see this in our example of Torrey pine, where Torrey pine is isolated. It does have low genetic variation, and it's been stressed for many years under water limited conditions. We see that the 5-spined engraver beetle has really got hold of Torrey pine populations because I think the trees were already stressed to some degree, and they've taken out so many of the mainland mature individuals, so that there's very few individuals contributing to future generations in that population today. So in other species, I've seen evidence of the exacerbation of the pest alongside climate change. But I'm not aware of studies that have shown it to date in ash.
Thanks, Jill. Merci beaucoup. Des questions orales. Jusqu'à date, on n'a pas de questions écrites. Et si vous avez des questions.
I have another one. It's going to be my last one because everybody's going to be tired.
Okay.
I would like to hear you about interspecific hybridization because in conservation, sometimes hybridization is seen as something like as a bad. There's a negative perception, but I would like to hear you about hybridization in terms of evolutionary potential, like the cross between different species, because yes, I want to hear you about that.
This is a very active area of our research program. We've been studying hybridization for a long time in a number of different species: Sitka spruce and white spruce hybrids, poplar hybrids, currently between Populus trichocarpa and Populus balsamifera. And we're combining whole genome resequencing data, multiple common gardens, to kind of assess, what is the role of hybridization to adaptation to climate change? And one of the things that I think we're seeing is that genetic variation that is maintained via natural hybridization provides admixed genotypes to persist in environments where their parental genotypes might not persist. So we do see evidence that admixture maybe can expand species range. So in the context of climate change, if considering seed transfer and seed transfer guidelines, it may be admixed genotypes that may be able to move more readily beyond the current range of a species. And I think that's natural hybridization that is occurring naturally, that's genetic variation in the wild that natural selection has selected to persist in those environments. So I think forest trees in particular have a long history of hybridizing naturally. We see this in oaks all the time, and we see it in spruce and pines. And I think so in forest trees in particular, I think there is opportunities to leverage natural hybridization as a new reservoir of genetic variation that may be able to allow populations to adapt to changing scenarios. We see some evidence that some admixed genotypes are more plastic than their parental genotypes across different environments. This is not uniformly true. There are some winners and losers in an admixed genotype, but we see some evidence that it can provide an advantage. And so I think there's a lot of value in natural hybridization, and particularly in the context of climate change.
Thank you.
Thank you, Jill. We still have time to have questions, oral or written. If you have an oral questions, please click on the raised hand and go to unmute mode. Donc, pour poser votre question orale, veuillez appuyer sur le symbole de la main levée en bas à droite et aller au mode et quitter le mode sourdine. Vous pouvez également poser votre question par écrit dans le chat box. OK, Nathalie, comme il n'y a personne, comme il n'y a pas de questions, donc
Yes, I think we have
go ahead.
actually, Jill is very busy, so I want to take the opportunity to ask her the question I have. So in terms of genomic vulnerability, I would like also to hear you about, because you present some genetic offset, like for the southern, but for the Fraxinus latifolia, and we've seen that, you know, southern population might be less vulnerable perhaps than the population from the north. But I would like to hear you about, do you think that using only genomic estimate, are you confident? Because there are also a lot of discussion in that approach.
Yep.
But how would you validate such a finding?
Yeah, that is a good question. I think there's a lot of debates in the approach. I think there are opportunities to leverage, if possible, common garden experiments to validate potential projections. But we're going to read Katie Lauderhouse's new paper this coming week in lab meeting. So I think I think the jury's still out. Genomic offsets have value, I think, in that they identify the climates and they might identify areas of vulnerability. Should we be using them right now for reestablishing our seed transfer guidelines? The challenge with genomic offsets is that they are only good as the data that you put into them. And I think there are still some limitations and a lack of an ability to really validate them yet. So I think we still have some work to do to operationalize the genomic offsets yet.
Okay. But at the same time, this isn't a type of information. There's a risk of doing nothing too. So we have to take I would say the less worst scenario.
Yeah. So the conservation biology is a crisis.
Yeah.
So can we use the information that we have? I mean, I'm fine with using them to point out vulnerabilities, and to maybe suggest maybe we can move some seeds into different locations. I just don't know that we can guarantee that all of a sudden you'll have fit populations if you move populations X into new environments.
Yeah.
Thank you. D'autres questions? On a encore quelques minutes pour une ou deux questions. orale ou écrite. So as I have the opportunity, I have a question, Jill, kind of basic science question regarding the polyploidy. You answered the first participant regarding the possible role of environmental constraints, but I think there's no evidence that the polyploidy is related to resilience, to environmental condition. But do you think that the appearance of polyploidy from a historical point of view could happen after a major climatic event? And I don't know if you have the tools to know what or when this polyploidy appeared. Is it 200 years, 500 years, more or less? Do you have information about that?
This is a really good question. So I'm going to defer to the fact that I have really great collaborators. Meg Staten down at University of Tennessee, Knoxville, and her PhD student, Zane, have been kind of going into the rabbit hole of the evolutionary history of the southwestern Fraxinus species complex with us for a little bit while, and Zane's actually interested in leveraging range-wide collections that we have for all of the North American ash species, and and we have a number of these collected and cytotyped already, so we know their polyploidy, and we're going to take a whole genome sequencing approach to assess the evolutionary relationships across the complex, and maybe we can identify when and how polyploidy occurred. Is it recurring repeatedly, or is it associated with one major event across the complex of species? But it's an open question in Fraxinus, which surprised me because, Fraxinus is a relatively common street tree. We see it planted in lots of different locations. And the fact that we don't know a lot about the evolutionary history of the species surprised me. But we're going to dig into this more with a lot of whole genome sequencing data in the very near future. So stay tuned.
Thank you. Thank you so much, Jill, for this excellent presentation. Merci beaucoup pour cette excellente présentation. Genomics-driven monitoring for genetic conservation and breeding in forest trees. En terminant, je vous invite au prochain colloque qui aura lieu dans 2 semaines, le 21 mars 2024, et qui s'intitule « Développement de méthodes de rechange aux herbicides en partenariat avec les communautés autochtones ». La présentation sera donnée par Nelson Tifo, chercheur scientifique au Centre canadien sur la fibre de bois, Ressources naturelles Canada, et Isabelle Allen, forestière de projet avec Wakotowin Development. Surveillez également notre fil Twitter et LinkedIn pour en savoir plus. Merci de votre participation et bon après-midi. Thank you so much, Jill. Bye.
Thank you very much, Jill.
Good to see you, Natalie. Thank you all.